PhD at the and all throughout that time, he was studying fungi and systematics. He'll tell us a little bit more about that as well. And then he did his first postdoc at LSU in a different topic on HIV forensics, and then moved over to plant pathology for an assistant professor position at Louisiana State University. And so today he's going to talk about the evolution of fungal pathogens across wild and agricultural habits.
Okay. Thanks, Sunshine, for the introduction. Thanks to all of you for showing up today. So I just want to start by saying my fundamental interest is in fungal biodiversity, and that's what motivates much of my research. Today, so a lot of the work in my lab centers on plant-associated fungi, but I also have projects working on animal-associated fungi like pneumocystis and attine ant cultivated fungi, and we also work on free-living fungi. So today, though, I'm going to talk about a couple different projects on plant-associated fungi that are taking place here, predominantly here in Louisiana. Before I get started on that, though, I want to talk about some of the different plant-associated fungi, lifestyles in fungi that we see, and offer a little bit of my perspective on how I think that we can improve our understanding of the life history of fungal plant pathogens by expanding our viewpoint beyond just that pathogenic life cycle or lifestyle and beyond agricultural habitats. So fungi have both an ancient and diverse set of associations with land plants. Saprophytic fungi, for example, are responsible for the breakdown of detritus in the form of plant biomass and therefore play an important role in nutrient cycling. Ecto and endomycorrhizal fungi play important roles in the acquisition of macro and micronutrients and water, for example, and have been shown to play or are thought to have played a role in the transition of plants from aquatic ecosystems to terrestrial ecosystems where most of the plant diversity is today. Plant pathogens, fungi are the most diverse group of plant pathogens, and they occur on all crop plants globally and therefore have major economic impacts. They also, though, occur in wild ecosystems as plant pathogens and have been shown to modulate plant diversity, particularly in tropical ecosystems. Thinking about the Jansen-Connell hypothesis. Endophytic fungi, there's not as much a large body of research on endophytic fungi, although that's certainly growing. These are fungi that colonize host plants but don't induce any kind of response and therefore don't make themselves evident. But they have been shown to play a role in host defenses and mitigate against biotic and abiotic stresses. But the lifestyles that I'm talking about here are not hard boundaries, right? So fungi exist and oftentimes sort of on a continuum across these lifestyles. So for example, saprophytic fungi have been shown to exist as saprophytes and be able to transition to become pathogens. And similarly, endophytic fungi can do the same thing. Now, certainly not all endophytes transition to become pathogens, but there is a large number of them that may do this and a large number that have been demonstrated to do so. And these transitions can occur in the other direction as well. Now, over the last, really mostly I would say the last 4 decades, since the work of Petrini and others in the 80s and then up to present, we've learned that endophytes are a very diverse group of organisms. Now, they're primarily composed of the Ascomycota, the phylum Ascomycota of the fungi, but they also include basidiomycetes and some lower fungi. And this figure on the top left is actually from a paper of Sunshine's, where they show just how diverse these endophytes can be in foliar tissue. So see, each one of those symbols represents a distinct morphotype, and you can see in just a 2 by 2 millimeter section the diversity of endophytes that you might find. And through research of Sunshine and Keith, for example, we've learned a lot about we've improved our biological understanding of what these organisms are doing. And as I mentioned, they've been shown to mitigate against abiotic and biotic stresses, increase plant fitness, etc. But a lot of these endophytes, unfortunately, have been placed in a rather coarse phylogenetic context. So the nuclear ribosomal ITS region has been chosen as the barcode locus for fungi, but it doesn't do a very good job actually at discriminating species, particularly in the Ascomycota. Within some genera, does a fine job, and others not a good job at all. In contrast to this are the plant pathogens. And the plant pathogens, there's a large body of research that is more than a century and a half here in North America, longer elsewhere. But a lot of that research tends to be habitat and lifestyle restricted, right? We're focused on agricultural habitats because that's where the economic impacts are. We're focused on the pathogenic lifestyle because, again, that's where the impact is. And also, it's a whole lot easier to collect a plant pathogen than it is to find a particular species that you're looking for as an endophyte because they don't make themselves known. In terms of phylogenetic context, a lot of plant pathogens surprisingly have not been put in a very robust phylogenetic context. And historically, the delimitation of species within plant pathogens has relied on this assumption that they're host specific. And so a lot of the species that we see today have been delimited historically, maybe partially based on morphology, but a lot based on host associations. So it's in this context that I'm going to talk about, and I'm sorry, let me step back to say that I think bridging the gap between these two lifestyles and starting to kind of expand our sampling into agricultural habitats and or outside of agricultural habitats, it can tell us a lot more about fungi that are endophytes and fungi that are pathogens. So it's in this context that I'm going to talk about a couple different projects that we've been working on over the last several years in my lab. One of those is, and both of these are pathogens of soybean. Cercospora leaf blight has been a persistent problem for soybean growers, particularly in North America, but also in South America. And I'm going to talk about sort of what we've learned about the biology and the species that are involved in this disease over the last several years. And then I want to talk about an emerging disease it's called taproot decline. And really, when I started working on this, we knew virtually nothing about it. And that was only 4 or 5 years ago. And so I'll start talking about that. And you know, a lot of the questions, I just want to say now, a lot of the questions that we think about with respect to taproot decline are the same questions that people have been asking for the last 2 years about SARS-CoV-2, right? So the kinds of questions that you ask with an emerging disease are the same for in a lot of ways, whether you're looking at a plant pathogen or a human pathogen. So I'm going to start with talking about Cercospora leaf blight. And Cercospora is a diverse genus in the Ascomycota. So the Ascomycota is the most diverse phylum in the kingdom fungi. And Cercospora belongs to the most diverse class within the Ascomycota, the Diphthidiomycetes. The Diphthidiomycetes have more than 1,300 genera and more than 19,000 species. And they reproduce sexually, if they reproduce sexually, through the production of pseudothecia typically, and asexually through the production of conidia. And there's a single conidium of Cercospora there. But not all of them reproduce sexually. So species delimitation in Cercospora, like a lot of other fungal plant pathogens, has historically relied on this assumption of host association. So, for example, Cercospora racqueticola on peanut, Cercospora hydrangea on hydrangea, and so on. And in a large part, host association was relied on. Part of, that was just the paradigm, but also there's not a lot of morphology within this group to differentiate species, right? So, Chuck in 1954 was the first one to do any kind of systematic revision on this group. And he recognized over 1,400 species, largely on the basis of host association as well, although he did do some synonymization. Ann Pollock in 1987 determined there are about 3,000 names in this genus, and then Krause and Braun in 2003 segregated out some of these species and were left with about 659 accepted species today. But most of these are only represented by type collections. So somebody went out there and they described this new species, deposited a type in a herbarium, and nobody's collected it since. Now, this paper in 2013 by Grenwald and others really changed the landscape for Cercospora systematics, because they put together a large collection of isolates from international culture collections and their own collections, sequenced multiple loci and put all of these into a phylogeny, and were able to start assigning names to clades. And this really provided the baseline data for others to start looking at the diversity of Cercospora in the particular systems that they were working on. And up to this point, it was really not possible to do that. So moving in back to Cercospora leaf blight, Cercospora leaf blight and purple seed stain, which is another manifestation of this disease, has major impacts wherever, particularly in the Gulf South or in the mid-southern United States. In Louisiana in 2018 alone, for example, it's thought that soybean producers lost approximately $22 million due to yield suppression from this disease. You can see in the top left, it causes these foliar symptoms. It's bronzing, purplish appearance early on in development or sort of mid-development, and then it eventually turns into this blighted, ashy appearance that you see in the top right. And that's where we can sometimes find Canidia being produced. It can lead to massive defoliation. This is kind of a very extreme example in the bottom left. And it does colonize the seed coat and causes this purple staining, although we can also isolate that from asymptomatic seed. So for really up until we started working on this, Cercospora cucucii was considered the causal agent of Cercospora leaf blight and purple seed stain. This was a pathogen that was described in the 1920s from Japan, recognized shortly thereafter in the United States, based on the same host and the same set of symptoms. And that was just the dogma. This, you see these symptoms on soybean, it's caused by this species. So, but we wanted to look at this question of what is actually present here on Cercospora leaf blight in Louisiana, and why look at this. Well, first of all, we know that cryptic species exist within individual morphotypes throughout the fungi. These are species that they're not, two different lineages aren't sharing genetic information, but they look just like one another. We also know that host specificity, or this assumption of host association, is unreliable for taxonomy. There's been observation of year by location interactions with regard to disease incidence, which led some to speculate about, there's more diversity out there, and also some documented population structure, although that was before we really knew, that there might be, or any consideration that there might be multiple species. The last thing is that fungicide resistance appeared really quickly in Louisiana, despite the fact that this disease has been around for a long time and people have been applying fungicides for a long time. It seemed to kind of happen over a year, and then it's expanded and is widespread in the state. So first thing we did, we drew on some collections that were already present at LSU, and then we collected more in 2013. We sequenced these 5 loci, which were the same loci that were sequenced by Grenwald. Right? So we looked at multi-locus sequencing of 205 isolates and placed them in that backbone phylogeny. And what we found was that there's 2 species of Cercospora associated with Cercospora leaf blight in Louisiana, and neither of those are Cercospora cucucii. So over 97% of the isolates are Cercospora flagellaris, and then there's a minority of Cercospora sigisbeckiae, no Cercospora cucucii. So what? So the reason that matters is that Cercospora cucucii has been isolated, and there have been multiple loci sequenced placed in a phylogeny. Every time that's done, and it's Cercospora kikuchii, it comes from soybean or a close relative of soybean, suggesting that kikuchii is potentially host specific. Well, that's important if you're thinking about a pathogen, because there's a whole lot of other hosts out there that might harbor this pathogen and contribute to disease on soybean if, in fact, we're looking at a pathogen that has a broad host range. And Cercospora flagellarius does have a broad host range just based on what has been sampled in the past, right? And here's a list of genera that all occur here in Louisiana of plants. Okay. So this raises the prospect that we're challenging another assumption, and that is that Cercospora leaf blight is primarily seed-borne. I showed you the seed infections, and this is kind of what people have been thinking, is that the reason that you get disease is because the fungus is present on the seed, it colonizes the plant as a latent pathogen or an endophyte, and then emerges as a pathogen later in the season. And there's evidence for that. We see, as I said, Cercospora is on the seed. There's been documentation of latent infection as early as B3, which is the stage at which there's 3 leaves on the plant. So that's early in development. And we do see simultaneous and uniform onset of symptoms often. So, but the fact that Cercospora flagellarius is a generalist kind of raised this question, well, maybe there's more going on than just seed-borne inoculum, and maybe seed-borne inoculum is not actually that important. So I want to look at this question and talk about how some approaches that we've taken to try to address this question. Now, one of the challenges is that people have not been able to reproduce the symptoms that you see in the field through artificial inoculation. And this has been a major challenge to try to develop, a program to manage these diseases. I have a student that's working on that now, and I think we're making advances, but that wasn't an option for us to look at this. So the first question that I wanted to look at to try to determine whether seed was the primary source of inoculum was to ask whether seed disinfestation would reduce disease incidence and severity. Simple. If you can remove the pathogen from the seed, then you shouldn't see any disease later in the season, right? If that's the primary source of inoculum for the disease. So what we did was we wanted a seed treatment protocol that would maximize the elimination of Cercospora, but obviously minimize any impacts to the developing plants, right? And so we had to come up with some way to disinfest the seed reliably, and it had to get under the seed coat, because we know that's where it is. So we first tested out using chlorine gas, because this is something that others have shown in plant pathology literature works. So what we did is we generate chlorine gas in this glorified aquarium in a chamber there with bleach and buffer. We were able to maintain chlorine concentrations in that chamber at about 200 parts per million, which we measured continuously. And then we then took those seed and looked at germination and measured seedling lengths as a proxy for seedling vigor, and then tried to isolate microbes from those seeds across different time intervals up to 15 hours. You can see that there's really almost no impact on seed germination. We're at 96% germination after 15 hours of treatment. No negative impact on seedling vigor between 0 and 15 hours. And chlorine gas eliminates microbes in 95% of the seed after just 9 hours of treatment. So this is what we're going to go with. So the idea was to take the seed and treat it, disinfest it, and then plant it out in the field, and then look at disease incidence and severity. So soybean seed is not produced in the state of Louisiana. It's all produced outside of the state. That's where all the seed producers are. So we got seed lots of the same variety from multiple different locations, and I'll talk about the reason why we did that in a little bit. We treated the seed for 9 hours. We planted it out in 2 different locations, 2016 and 2017, treating over 4,000 seed per location per year per seed lot. And I'm just going to show 1 year of data because it looks the same, virtually the same in 2017 and across seed lots. We don't see any impact to disease incidence or disease severity resulting from the seed treatment. There wasn't that high of disease incidence or disease severity. You know, we got up to 20% disease incidence and up to 25% disease severity. Nonetheless, we don't see any differences between the chlorine treated in red and the untreated in blue. We also took seed from all these seed lots. We isolated Cercospora from them. We sequenced them with multiple loci and placed them in a phylogeny. And what we found was that there were 3 species on the seed, and that included the 2 that we'd already found, plus Cercospora cucucci. So Cercospora cucucci is out there, but it's just not a big player, at least in Louisiana. But this also raised the question, maybe we can look at if there's any changes in community composition from the time of planting to the time of the emergence of the disease on leaves. So what we wanted to do was just look at sample seed lots prior to planting, isolate all of the cercosporoid fungi, and then assign them to species, sample from symptomatic leaves several months later, and then characterize that community. We take it all the way to harvest, we collect the seed, and then we isolate and characterize that community. Now, these aren't real data. This is what we were kind of expecting to see. And our expectation would be that if this was all vertically transmitted, meaning from the seed to the leaves, then you would expect the same community composition on leaves as you see on the seed that you planted. Right? So, for example, the proportion of isolates that belong to Cercospora flagellaris in brown there, at 35% on planted seed would be about the same at the time of looking at blighted leaves. Okay, so this, what we in fact saw was a shift from the time of planting to the community on blighted leaves. And it's more pronounced on some seed lots and some years, but for example, this is the Tennessee seed lot. For 2016, you see that Cercospora flagellaris made up just 25% of the isolates on the seed. And, you know, again, these are all untreated seed. But by the time we get to blighted leaves, 85% of that community was Cercospora flagellaris. And we see a coincident reduction with Cercospora sigisbeckiae, where you have 55% of the seed are harboring sigisbeckiae. By the time you get to blighted leaves, it's less than 15%. Okay, and we see significant changes in 2017 for flagellaris, but less pronounced on the Tennessee seed lots, and also significant changes in the same direction from the Arkansas seed lots. Okay. So then the last thing that we wanted to look at was just to try to identify alternative host species for the disease in Louisiana and figure out whether those alternative hosts are actually contributing to the population. So it's one thing to find the pathogen on other hosts, but is it contributing genetic information to the population on soybean and vice versa? Is there gene flow between these populations? Now, this species, Cercospora flagellaris, which is the dominant pathogen, was described from this plant here, Phytolacca americana, in the late 1800s, 1880s, from Pennsylvania. You can see that the distribution of this plant overlaps certainly with the distribution of soybean in Louisiana, but really with the distribution of soybean everywhere except for the upper Midwest. So this is one host that we were looking for, but we surveyed lots of different host species, and we were just looking for cercosporoid fungi, cercospora-like fungi. And we isolated Cercospora from cotton and pokeweed, mulberry, giant ragweed, Triodonus, and other hosts. What you see here is this is Cercospora and giant ragweed. We think that that's Cercospora causing these symptoms that look similar to what you see on lighted leaves of soybean. This is Cercospora on Phytolacca. You see that it produces these more defined circular lesions. So what we did is we isolated all these species, we sequenced them, placed them in a phylogeny to assign them to species. And what we found was that multiple Cercospora species can occur on one host, and multiple hosts can harbor one species. Not particularly surprising, but what we did find was that Cercospora sigisbeckiae has a lot of overlap in the host, particularly with this alternative host, Phytolacca americana. So we see that on soybean, cotton, and pokeweed. With Cercospora flagellaris, it has a broader host range. We found it on soybean, cotton, mulberry, ragweed, pokeweed, etc., and on Cyperus, which is the only monocot in this group. And I suspect we keep searching, we keep finding it. So then the next thing that we wanted to do is determine, are these hosts actually contributing to the pathogen population, right? And we also wanted to look at if there's any evidence of dispersal across geographic areas, because we have this seed from multiple different locations. So what we did is we genotyped these isolates at over 19,000 SNP loci using a GBS approach, and took isolates from soybean, a collection of isolates from Phytolacca, included one isolate from Triodontis, and then we have representatives of Louisiana, Tennessee, Arkansas, and Illinois, although the sampling really is adequate in Arkansas and Louisiana for this question. What we found was that the isolates from Cercospora flagellaris from weeds around soybean fields do contribute to that population, right? They're part of the same population. So what you see here is a PCA plot, basically a two-dimensional representation of genetic differences among isolates based on those SNP loci. The distance between 2 points approximates their genetic distance. Each of these colors denotes a different host species. So soybean in green, Phytolacca in brown, and then there's that one Triodontis isolate in purple. And you can see that these, and some of them are obscured, but the Phytolacca isolates are nested right in with soybean up in the top part of this plot. These isolates are from Phytolacca as well, but these come from the eastern side of the Mississippi River, close to Baton Rouge, where there's no soybean being grown. So it seems like these from non-agricultural populations are, while outside of agricultural populations, but in close proximity to soybean plots, they are part of the same population. But you move away from those soybean growing areas, and it looks like it's a different population. But obviously, the sampling is too low to make very strong statements about that. But we did see that the levels of population structure between those agricultural and non-agricultural isolates is greater than what you see across geographical regions, right? So these are all isolates drawn from seed from these different states. And you see isolates from Arkansas, for example, are clustered with isolates from Louisiana. So it seems like, in this case, that there's something is moving this species across state boundaries, and maybe that is on seed, right? So maybe seed has played a role historically and still is playing a role, but it's certainly not the sole source of inoculum. So we saw seed disinfestation doesn't reduce disease. We see a consistent shift in the community from planting to the appearance of symptoms, and we see these species on multiple hosts, as well as ongoing dispersal between soybean and those other hosts. The fact that we found these isolates from non-agricultural populations that are extinct is not necessarily something that's directly useful for plant pathologists looking at implementing certain mitigation strategies. But this might be a useful resource to understand genes that are involved in adaptation to these environments or to this host. And we actually just got a grant from the USDA to look at this question across the southern United States. So now we're monitoring airborne dispersal. We've set up spore traps around the state. and developing qPCR probes to kind of figure out which species are present, when and where, and what abundance. And I have a collaborator who's trying to leverage this information to develop fungicide application timings that will be more effective. We've sequenced, we're, as I said, expanding collections from non-agricultural areas, and we've published the genome of Cercospora sigisbeckiae and Gucci and have about 20 other genomes that have been sequenced and we hope to publish soon. And then I have a PhD student who's trying to look at these different infection strategies of the different species to see if that might be why we're getting this shift from the time of planting to what appears on leaves and again on seed. So the next thing that I want to talk about is this emerging disease, taproot decline. And as I said, a lot of the questions about emerging diseases on plants are the same as emerging disease like SARS-CoV-2 or COVID-19. So we want to know what's the cause of this disease, how did it emerge in the southern United States, and what's the role of non-agricultural hosts in this disease? Is there a role there for its emergence? And then I'll also talk about briefly at the end some of the other questions that have come up as a result of trying to answer these questions. So taproot decline of soybean, really, it was around, people mentioned it, like agricultural consultants, for example, mentioned seeing it, but it was just kind of a mystery what caused it. Some people thought they knew what it was. Black root rot of soybeans used to be Chalabiopsis, Berkeliomyces. Sudden death syndrome, which is caused by Fusarium species, Fusarium frugaliforme, which actually produces metabolites in the fungus, and those get translocated to the leaves where you see the symptoms. That's also a root-borne pathogen. And nutrient deficiency. So all of these symptoms in the leaves look pretty similar, except for Chalabiopsis, where it's the root symptoms that are comparable to TRD. So this is what the symptoms look like a little more close up. So these appear can kill seedlings all the way up to mature plants that are producing beans. It causes this interveinal chlorosis, that yellowing, and necrosis, kills the leaf cells. But the pathogen we don't isolate from the leaves, we find it in the roots. And it causes this necrosis on the roots, but it also forms a black stroma around the roots. And you can see in the pith of the taproot, there's mycelium. And so you can pull these plants up. A lot of times the taproot breaks right at the crown. You split the roots down the middle, and then you can just transfer the mycelia right out of the pith onto a plate. And most of the time, and this is also interesting, is that 80% of the time you get no contaminants. Right? You only get taproot decline pathogen, which is surprising from this environment, a root environment. So I was worked with several pathologists to get this paper out to where we have identified what taproot decline is. And they were ready to push it out to describe the symptoms and just put a name on this thing so that other pathologists and other parts of the United States would be aware of it, and maybe we could get a better idea of what the actual distribution of this disease is. So we sequenced the genome of one isolate, and we didn't have a big collection of isolates at that time. So we were able to determine that this species, the taproot decline pathogen, is in the Xylaria arbuscula species complex. But we didn't put a name on it. And that's because it's on this long branch here, which suggests maybe this is a new species. But again, we only had a small number of isolates, and I wasn't really comfortable with describing a new species on that basis. So we wanted to look across the distribution of this disease. Now, what's interesting to note about Xylaria is that there's really few plant pathogens in the genus Xylaria. Now, in the family, Xylariaceae, there are plant pathogens. But there's about somewhere around 670 species in Xylaria. And the largest proportion of them are actually wood decomposers or saprophytes. There are a lot of endophytes that have been discovered, in papers published of endophytes in Xylariaceae from Yanni Arend's group in Arizona, particularly. But most of these are either endophytes or decomposers, not pathogens. Very few species. So the first thing we wanted to look at was just, a straightforward question, is this a novel species within the species complex? And then the second was, is there any indication that there's been a shift in lifestyle from being a saprophyte to becoming a pathogen within that species, right? So, as I said, most of these species are saprophytes. But so what we wanted to do to try to look at this is try to sample from non-agricultural areas, get out into forested ecosystems, and also to look in herbaria for historical specimens. As I said, it can be really challenging to find these pathogens when you're not sort of in a monoculture in an agricultural habitat. So what we did was we looked for these symptoms across the known range of the disease, right? So we sampled symptomatic plants from Alabama, Arkansas, Louisiana, Mississippi, and Tennessee. We looked for the root symptoms associated with them. We also collected these specimens on debris, which are sterile stromata of Xylaria, and we found it on corn, cotton, and soybean debris. Then we isolate the pathogen into pure cultures, and then that's what we use for sequencing the same loci that we used in the first phylogenetic analysis, which are loci that are available for Xylaria in public databases. Then we sampled U.S. herbaria looking for specimens that were in the species complex, but also overlapped in distribution with the taproot decline pathogen. And we drew on several collections from the U.S. National Fungus Collection. Jack Rogers, who recently passed away, who spent his career working on Xylaria, he sent us several specimens. And then we also found specimens in the LSU herbarium of Xylaria arbuscula. And we also looked just searching for databases. Once we had a few loci sequenced, we could look, for example, on GenBank and look in the literature. And we found this one specimen from Fournier et al. In 2020, where they described morphology of this thing, but they only had one collection of it from Martinique. And they said, this is probably a new species, but we don't know what it is. And then we found this whole genome sequence deposited from Xylaria striata, which was a sister species in our first analysis. And so they called it Xylaria striata. And this was an endophyte of wild rice in China. Everything else that we had was in the Western Hemisphere. So what we found collecting these pathogenic isolates is that, all of these they all share a most recent common ancestor, all of the pathogens. You can see those in green. We had sampled across all of these states. You see the distribution map there. So we knew, the pathogen is the same species across the distribution. And it does look like that it is a novel species. Okay? So we still see the sister group relationship to Xylaria striata. There are some morphological differences between these species. And we're fairly confident that we are confident this is a new species and described it as Xylaria necrophora. This specimen from Martinique, which was collected as a saprophyte from the forest floor and outside the range of taproot decline, is also nested within this clade. And so that was included in the description of Xylaria necrophora. We also see these historical specimens from Florida in the 1920s, and these came from predominantly oak forests, and this were probably, we don't know what the substrate is that they're on, but it is a woody substrate and was probably a saprophyte. And then we found these specimens in the LSU herbarium, which also nested within Xylurian acrophora, these were deposited in the 1930s and 1940s. One was from sugarcane debris. Wasn't noted as a pathogen, and the sugarcane pathologist has never seen Xylaria on sugarcane. So we don't really know. That could have been a saprophyte. Who knows what it was doing there? But it was on sugarcane debris, and then one that was on a gardenia, wood of gardenia. And we don't really know anything about it. There's a date on it and a location when it was deposited in the herbarium. And these are, of course, within the range of the disease. So based on these, Xylurian acrophora is a novel species within this species complex. And based on this sampling, it appears that there's been some kind of shift from saprophyte, Venus saprophyte in non-agricultural areas to a pathogen on soybean. And soybean hasn't always been present in the southern United States. So it's introduced. And so it was originally grown as a forage crop, and we probably, a lot of forests were cut down in order to start growing soybeans. And so it could be that this was present in the soil and emerged as a pathogen. Why it emerged recently, who knows? Maybe it's because there's some genetic base in the chain in the cultivars that are being used. For example, glyphosate resistance was introduced around 2000 in soybeans. And maybe that had something to do with it. Or you just get a single introduction in a soybean and then it spreads from there. So that's a question that we wanted to look at, try to figure out, does this have the patterns, genetic patterns of a novel introduced pathogen, or does it have patterns of one that's endemic and just emerged in multiple places at the same time? So I'm not going to talk in too much detail about this part just in the interest of time, but I had a PhD student who's just finished up work on this. We sequenced whole genomes of 170 isolates of Xylaria arbuscula and Xylurian acrophora, including some of those herbarium specimens, but we didn't get great data from the older specimens. And we wanted to look at population structure and try to identify center of diversity for this pathogen. What we would expect to see if this was an emerging pathogen, emerging in multiple places at the same time, and that it was endemic and then emerged, is significant population structure across the geographic area of the disease. If there was maybe a single introduction and then spread from there, you wouldn't expect to see population structure across geographical areas. And in fact, in soybean, we don't see any geographical population structure. We do see some population structure without any a priori population assignment. It seems like there's two lineages in this population, but they're not that different from one another. And so we think this is probably was a single introduction. And although our sampling is biased towards Louisiana, we see the most diversity here in Louisiana. So it may be that it was introduced here. We're also looking at, trying to make inferences about whether sexual recombination is occurring, because we do see stroma, but we don't see ascospores or sexual spores. And what we've inferred from just looking at linkage disequilibrium across the genome and then simulating linkage disequilibrium under purely sexual population, mostly clonal, up to clonal, is that these lineages are probably mostly clonal, right? And the signature of recombination is probably a historical signature of recombination rather than due to ongoing sexual recombination. Now, I've got an undergrad who's working on using these genome sequences to develop qPCR primers as diagnostic tools, but also to look at the distribution of this disease. Actually, after we published the new species paper, I caught email from somebody at Bayer who said, hey, we've got soil collections from all over the world. And if you can find this thing, if you can develop species-specific primers, we can just go through and figure out what's the distribution of this based on our soil sample. Right? So I've got an undergrad who's working on that as part of her honors thesis. and we'll see where that goes. The other thing, I had a master's student who just finished up also. We're looking at trying to understand, well, how does this pathogen cause the symptoms that we see? So something I didn't mention about Xylaria is that this genus produces a lot of bioactive secondary metabolites. And so that's one of the first things that I thought of when we finally identified the pathogen. Well, we need to start looking at the role of secondary metabolites in this infection. So what we did was look at the impact of cell-free culture filtrates. So we get rid of all the mycelium and just look at the secondary metabolites that the fungus produced and looked at their impact on soybean plants. And we're able to reproduce the symptoms just with the secondary metabolites. And we also see that it majorly inhibits root growth. And you can see this is after they've been exposed to metabolites. Control on the left. All these black cells are dead cells. And we also found that probably the reason that we only or very rarely recover other species from roots is that these secondary metabolites inhibit nearly everything that we've put up against it. And we've now identified, there's a lot of metabolites that we haven't identified, but we have identified a couple of them that have been shown to be cytotoxic. Cytocalasin C and cytocalasin D are produced in high abundance by this fungus. And so this is probably what's responsible for the symptoms or for the infection. We don't know that these metabolites are being translocated in the leaves, but they certainly play a role in disease. So with that, I'll just leave it there. There are lots of people to thank, but the people highlighted in yellow are the ones from my lab that have worked on these two projects. I'll be happy to take any questions.
Haley has her hand up.
Where?
Oh, is that
Oh.
I'm interested in better understanding how, again, forgive me because I'm not a mycologist.
Yeah, that's fine.
But I'm thinking about how, you have these microbial communities that are diverse and exist pretty ubiquitously all over the place. So, do you have a sense that these are prevalent or these are present, but the virulence, something kind of kicks up the virulence or the effect of the disease or the effect of these metabolites? And could that be related to, temperature change or things like that?
Yeah, I almost told you about another undergrad project that we've got going on with this, but I'll also talk about Cercospora leaf blight with that question. One of the things so these cytocalasins are nitrogenous compounds. And so one of the things that came into my mind first is, soybeans often grown in rotation with corn. Soybean has nitrogen fixing rhizobia, right? But corn doesn't, and it takes a really high nitrogen input. And so one of the things I'm thinking is, maybe the high nitrogen applications that you put down when you're growing corn actually sort of promote the production of these secondary metabolites that then you go in and you put soybeans in and they're ready to kill them, because we've also looked at these metabolites. We haven't looked at their impact on corn, but we have looked at their impact on other host plants. And for some reason, they seem to be impacting soybean uniquely. But we know they impact other hosts. It's just not ones that we've looked at that are grown in rotation with soybeans. So we're looking at that, but we're also going to look at impacts of temperature changes, sort of thinking towards climate change. But so we're going to just monitor the production. We've got standards for these, internal standards, so we can look at how these different growth parameters impact the production of cytocalasins. And so there might be something there. The other thing with Cercospora leaf blight, I mentioned that this has been a disease that we really haven't been able to reproduce in the greenhouse or in the lab or even artificially in the field. And it's just been a huge struggle. Well, the first struggle was getting to produce spores in artificial culture. Nobody could do it. And then we tried out a bunch of methods. We've got a reliable means of generating spores in culture now. But then we'd go in and spray plants, and we just couldn't get any symptoms. And I mean, I don't have a lot of experience with it, but we'd send spores to collaborators that do, and they weren't getting symptoms. But I have a student from Brazil who got his master's in plant pathology, and we started talking about it, and we decided to look at what I call sort of priming or acclimatizing the plants before you do an inoculation. So we looked at 25 degrees, growing soybean plants at 25 degrees for 4 or 5 days before you inoculate, growing them at 30 and growing them at 35. When we grow them at 35, we can reproduce the disease symptoms consistently, and they're pretty severe. At 25, they're almost absent. So there is a role, right? And there is something that is happening out there that might be causing this shift. But with taproot decline, I mean, I mentioned these glyphosate resistant soybeans, that everybody knows about, right? Roundup Ready soybeans. I mean, those came on the market around 2000, which is around the same time that this disease seems to have emerged. So that's another thing that might have happened to contribute to this. Yeah, so I'm trying to keep good.
Okay, first I have a comment. I think it must be really fun to say Kikuchiite. You get to say that word a lot. That must be fun. My question though is, when you are looking for alternative hosts for these pathogens, how do you decide where to look?
That's a good
How do you make those decisions?
Well, I mean, in some ways I try to not come up with some preconceived idea of what is going to be the best place to look. And try, botanists like to say, go out and bale hay, when they just go out and collect everything, and that's kind of at the first, I mean, we were obviously looking close to soybean fields initially, and then we were looking on Phytolacca because that's where it was described from. But other than that, it's kind of like, we'll go out there and look for lesions on plants and pick them up, or don't look for lesions and just isolate. So when I did my PhD, I was doing this. I was looking for this endophyte in native cranberry bogs. And I mean, people used to walk through the lab and I'd have just plates stacked up like this, on the lab bench. And they're like, what are you doing? Oh, well, I went out collecting last week, and I collected all these plants, and then I spend 3 weeks to a month plating them out, endophytes, and then looking through the microscope trying to find this thing, and it's like, what'd you get? Oh, I found one. I mean, that's just the way it is, right? If we want to find out what these organisms are doing out there in the broader world, then we kind of have to look everywhere in a sense. I mean, some of it can be guided, but I don't, So that's kind of the approach we've taken.
Yeah, I was wondering, with this taproot decline pathogen, it's in the roots and you can see the mycelium, what have you. I think when the soybeans harvested, the roots must remain in the soil pretty much. So do the fields become progressively less viable for soybean cultivation? Or if there's rotation, does that take it back to the starting point?
Yeah, particularly since, or at least this is kind of a hypothesis of one of my collaborators who's he's out, does extension pathology, is that, so there's been this sort of advent of precision agriculture using GPS coordinates to plant in the same location year after year after year. And he thinks that actually that precision agriculture is placing those soybean seeds right next to last year's debris. And that's where the infection's coming from. So that's one of the other things that they've done. We can't do it in real time, but like with nematology, they've used electrical conductivity to try to predict where certain nematodes are in the field. What I want to do with this qPCR primers is to just go out and sort of sample soil on a grid. And then we can say, hey, this pathogen's not over here. So either if you care about it, stop driving your tractor from here to there. And you can plant whatever soybean variety here, but you need to plant resistant soybean varieties here. But yeah, absolutely. I mean, I think that's the source of an infection most likely. But how is it moving so quickly across this broad geographic area? I don't know that.